We still have some folks coming in, but we have quite a lot to cover and we've gotten kind of behind, as you guys have seen. So I'm going to try and get started here. Before we begin, there have been a couple of questions about Wednesday's summaries that I thought I would share the answers to with everyone. So one question was regarding, do you need to put the question that I require in the text? And yes, you do. Okay, so please include that in your summary. If that is on the second page, that's not a problem. Okay. Um, and then the other question that Hamza asked today was the discussion points, the 5 points. Um, where does that come from? And that's from the in-class participation on Wednesday. So you need to ask a question or answer a question or participate in some way in the chat or verbally during Wednesday's class to get those points. Okay, and that's a part of your discussion. That's a part of your 25-point grade for the summaries. Are there any other questions about the summaries before we begin? All right, so let me share. So we left off switching from the aquatic environment to the freshwater environment to the pelagic environment or the oceanic. And so compared to, we mentioned that there were higher nutrients in the near shore compared to the open ocean, where the nutrient levels are often quite a deal lower. We do have a similar term in the epipelagic, like we had the epilimnion, which is the mixed layer. It's also often high in light, and so it's sometimes referred to as the photic zone. And that's where light is going to be penetrating in terms of serving its important role. It'll also be warmer, of course. And what you can see on this diagram to the right is that we can have a thermocline, or another type of gradient or cline is the pycnocline. And this is a salinity gradient or a density gradient. So it may be a thermal gradient, or it may be due to the change in salinity with depth. Okay. And then as you get deeper and deeper, of course, it gets very low temperatures. It never really gets down to zero, of course, because it's salt water, but it approaches about 1 degree in the real deep depths in the bathypelagic and then in the hadal zones. Okay. There's, of course, no true photosynthesis down here. Although I've once read an interesting paper about photosynthetic organisms that live near hydrothermal vents and use the infrared radiation for their photosynthesis. But in general, we can ignore that. So it's very cold. The other thing that'll come into play today in our extremophiles discussion is high pressure. So the pressure is very high as you go down in the depths of the ocean. So speaking of going down in the depths of the ocean, one of the important components, even though this is a water environment, it turns out that it's not just water. Particles are very important. They're not as important as they were in the soil and sediments, but they're still very abundant and important. And these are typically aggregates that include cells as well as extracellular polymeric substances, or EPS, which is kind of a mucousy polysaccharide, sugar-rich excretion that is released by microbes into the environment and then serves as a way of binding together or aggregating cells and often like things like the decaying frustules of a diatom. That's like the skeletons of diatoms, things like that. So aggregation becomes important. And in many cases, under certain circumstances, this is actually visible to the naked eye, and we call it marine snow. And so there are some, you can just Google marine snow and you'll see images where it's really dramatic. Chemotaxis is very important when it comes to marine snow. In terms of the microbes finding these aggregates. And I'm going to show you a picture of that in a minute. The other thing is this is a very large export mechanism. So most of the productivity in the ocean is going to occur where? Is it going to be near the surface or down deep, do you think?
Surface.
Right. And so if that's where the primary productivity is, what is it that feeds the deep ocean? Well, it turns out that this kind of marine snow or other exported things like whale falls and other exports of things that are normally living in the surface and fall down can be really important in bringing carbon and nutrients. And so that's shown here on this image. So the discovery of marine snow is really attributed to a Scripps UCSD researcher, Farouk Azam, who identified this, as you can see now, 20 years ago. And what he and his colleagues demonstrated was that this export function, was really important. And what you can see is that as the marine snow particle trails down, it leaves a tail behind it, and that's shown in green here. So we have dissolved organic material, um, things like carbon, nitrogen, that are, of course, very important, will be kind of streaming off the back, almost like a comet's tail, as this particle falls down in the water column. And so this becomes this export function, but it also attracts the chemotactic microbes to it. And so you end up with like a whole little ecosystem on marine snow where you've got um, primary producers, you've got heterotrophs that live there, and then you get grazers or things that will be attracted to it to eat the bacteria or the algae that are living on this marine snow. And then you can have viruses infecting those cells as well. So it becomes its own little food web in a sense. And we'll come back to sort of the marine food web at the end of this lecture as well. So who is it that lives in aquatic habitats? Well, as we said, it's almost always going to be gram negatives, but unlike in soils, you don't really have a large abundance of gram positives, or a relatively large. So the gram negatives really dominate in aquatic environments in both freshwater and in the ocean. So you'll get a number of different groups. Things like Pseudomonas or Colobacter can be very common in freshwater. Colobacter is kind of interesting because they grow these stalk-like structures which allow them to get up off the bottom of the lake that they're living in, and that allows them to be in more flowing water and get away from some of the diffusion issues that we've talked about. Uh, there's a number of groups like Vibrio that are very common in both freshwater and especially in the ocean. There are phototrophs. In addition to cyanobacteria, there are also phototrophs that do not produce oxygen, like the purple and green sulfur bacteria. And nitrogen cycling, as it is pretty much everywhere, is very important. So one of the things we discovered as we started to sequence 16S ribosomal RNA genes from the ocean in the 80s was that there was a large abundance of microbes there that were not cultured at that time. And so one of those early studies that was done um, by Steve Giovannoni's lab at Oregon State, they went out to the middle of the Atlantic to an area called the Sargasso Sea, which is so named because there's this sargassum weed that's prevalent out there. So this is very low nutrient open ocean water. And they did sequencing of the 16S gene and identified different groups, and they just numbered them SAR1, 2, 3, etc. And it turned out that SAR11, which at that point was not cultured, um, was found everywhere in the world's oceans. And in fact, it's one of the most commonly identified organisms on our planet. And they, through many years of dutiful research, finally were able to culture one representative called, and they named it Pelagibacter for the pelagic zone. Archaea are also very important there. Originally, they were identified the marine Kren archaeota, And now this group has been sort of expanded and has been renamed the Taum Archaeota. Many of these are nitrogen cyclers, and the lab that I did my postdoctoral research in at the University of Washington was the first to isolate one of these nitrogen cyclers. This is an ammonia oxidizing organism called Nitrosopumilis. And this was an important organism to discover because when modeling of the nitrogen cycle in the ocean had been done prior to this, there was always missing nitrogen. And so trying to figure out where some of this nitrogen was leaving was an important area of research. And it turns out that these archaea were responsible for converting the ammonia to nitrate, and then denitrifying bacteria and archaea converted that to N2 gas, and that's why it was being lost. In addition to bacteria and archaea, of course, fungi and protists are really important. I mentioned flagellated organisms earlier as being important with marine snow, but they are very prevalent predatory organisms in addition to ciliates, which you guys may be more familiar with. And they're often quite small. So the flagellates, some of them are small enough to be called nanoflagellates. And what they do is they graze on bacteria directly, so they can lyse those bacteria. And as I mentioned as well, viruses also infect all of these microbes that live in aquatic habitats and play an important role in releasing carbon and nitrogen back into the environment when they lyse the cells. We're going to talk much more about viruses a little later date in the semester. So I've alluded to this a bit, but this graph just or figure just kind of shows you where there are different zones of organisms that are found in this, let's say, freshwater lake. So at the surface, as we've talked about, cyanobacteria or other oxygenic phototrophs would be important. But as I mentioned, there are some microbes that do not produce oxygen in photosynthesis, and they are often found a little bit deeper. Now, the oxygen that's produced here, as well as the oxygen that gets mixed in from the air, is going to drive aerobic organisms, respiratory organisms. Okay, but down in the bottom of the lakes, and particularly in the sediments, but you can actually have anoxic water at the bottom of lakes if they're not well mixed. And when, again, would we have not well mixed water at the bottom of lakes? Do you guys remember?
During the summer months.
That's right. And why was that?
I'm sorry.
Why was that? You're
Because
correct.
because
So,
there's no thermal equilibrium during that
right,
time, and there's higher water temperature as compared to the bottom.
right. So it was the thermocline or the stratification that got established. And so when you have that, remember, oftentimes the water down in the hypolimnion is low in oxygen, and it can even be anaerobic. So these anaerobes are very commonly found down there. And so you can get a mixture of methanogenic, and this is a typo. This is an old diagram. I apologize. This should be archaea. There are no methanogenic bacteria. So this is a misnomer. So methanogens and other organisms like sulfate reducers and denitrifiers and fermenters become very important. When there's high sulfate, like there is in the ocean, sulfate reducing bacteria tend to dominate. And when there's low sulfate that may be limiting for this group, that's when the methanogens tend to dominate in these habitats. So in freshwater habitats, it's thought that methanogenesis is a more important process. And in marine environments, sulfate reduction. That said, there are methanogens in the ocean and there are sulfate reducers in freshwater. So it's not 100%. Another term that we use for the bottom, either of a lake or the ocean, is the benthos or benthic organisms. So these would be organisms that are in the bottom, okay, as opposed to what was that term that we talked about in terms of the organisms floating around?
Planktonic.
Yeah. Okay, so benthic is kind of the opposite of planktonic. So as we discovered the importance of both bacteria and archaea in the ocean, um, researchers went around the world and they counted up using specific approaches to count genotypes of bacteria separate from archaea called FISH, or fluorescent in situ hybridization. We're going to talk about that in a few weeks. And they used this method. And what they found is that if you look on this, this is the percent of the total count. And these are, orders of magnitude sort of log numbers, right? So at the surface, you get 10 times as many bacteria as you have archaea. And so that's why they make up so much more here. But what do you notice about this graph as it goes down?
The bacteria decreases and the archaea increases.
Exactly. So what you'll see is that as you get all the way to the bottom, you end up with roughly equal amounts. Excuse me, sorry. Let me get my mouse sort of situated. So what you can find is they actually both decrease. So this is the relative percent. What you can see is this goes from 10 to the fourth to 10 to the third. This goes from 10 to the fifth to 10 to the third. So the decrease in bacteria is an order of magnitude higher. And so that's why you end up with about 50-50 distribution, even though you started off with more bacteria at the surface. And so archaea and bacteria are both important, but the archaea really shine, so to speak, or are really important down below about 100 meters. Incidentally, this 100-meter mark is often associated with that photic zone that I was talking about in terms of the penetration of light. Below 100 meters, it gets very dark. And that's in the open ocean. In shallower or coastal waters, the photic zone often doesn't penetrate as far. All right, so let's have a poll question. Let me get this going. So which of the following about oceanic habitats would be true based on what I've just told you? Aside from phototrophs, Crenarchaeota are the most abundant group of surface microbes. Particles are very important for keeping nutrients near the surface. Sulfate reduction is the major anaerobic metabolism in ocean sediments. Or D, open ocean habitats have more nutrients than coastal habitats. So I'll go ahead and launch the poll. Which of those four is the best answer? Read the questions closely and carefully. Some of them are a little tricky. Okay, let's go ahead and just pick an answer and then we'll talk about it. All right, is that everybody? Okay. So it looks like the majority of you put C, but quite a few put B. So let's take a look at it. So nobody put A, so that's good. Because we just learned that Crenarchaeota are not the most abundant group of surface microbes. Where do they become abundant? Where did we see the increase in archaea?
At the bottom.
Yeah, so in deeper waters. So you guys all got that. Okay, so here's B. Why is B not the right answer? It seems like a good answer, but why is it wrong? Maybe somebody who put C and figured out that B wasn't correct. particles are very important, right? But do they keep nutrients near the surface?
No, they're going to float downward or drift downward.
That's right. So they actually, we said, were a export function, right? So that's why B is not the correct answer. So if it had said they're very important exporting nutrients to the floor, the seafloor bottom, that would be the correct answer. So sulfate reduction is the major anaerobic metabolism in oceanic sediments, and that's because sulfate is very abundant in the ocean. So the ocean is salty, but it's not just sodium and chloride. It has a range of salts, including sulfate. All right, so does everybody understand why C is the correct answer and B is not at this point? That was a tricky one, so don't feel bad. Hopefully now that you have been caught up with that, now you'll remember. All right, so we've talked about the significance of photoautotrophy or photosynthesis. And so I just want to kind of tie a bow around this and give you a general sense. So it's thought that about 50% of the world's carbon fixation or primary production occurs in the oceans, right? The other half is going to be on land and is going to be dominated by terrestrial plants. But in the ocean, most photosynthesis is occurring with microbes, either cyanobacteria and photosynthetic bacteria, or eukaryotic algae like diatoms and dinoflagellates and chlorophytes and things like that. And of course, this is why photosynthesis is important near the surface, because there's light, right? And so we call it that euphotic zone or true light zone, okay? Now I mentioned that light decreases with depth. And so the bottom of the photic zone kind of is roughly a point called the compensation point, and that's at about the 1% light level. That's where photosynthesis is going to be balanced by respiration. Above that, near the surface, there's more photosynthesis than respiration, and below that, respiration rates will exceed photosynthetic rates. Okay? So just in case you needed more terminology, we also have some of here. So for lakes, the bottom of lakes, particularly deep lakes, would be called the profundal zone. And then in the ocean, it's the bathyal or the hadal zone, depending on how far down it goes. Okay. Not much in the way of photosynthesis going on in either the profundal zone or the bathyal zone. Okay. All right. So algal blooms. We've talked about eutrophication and nutrients that lead to the proliferation of algae, especially eukaryotic algae that form blooms, like dinoflagellates that we've talked about. But you can definitely have blooms of things like cyanobacteria. And in fact, they often produce blooms in freshwater environments. So one of the things that leads to algal blooms appearing near the surface is there are buoyancy mechanisms. So cyanobacteria produce something called gas vesicles, which are these protein structures. They're not like little balloons, but they basically are structures that proliferate and then they have gas in them. So they cause it to be buoyant. And when conditions are calm, it can appear as if there's like a bloom that formed overnight. We as biologists understand that, things don't, replicate so fast that it might happen overnight that you would get billions of cells. But what happens is they may be replicating and replicating a little bit deeper down. And then if you have a calm night, the buoyancy mechanisms bring everything up to the surface. And oftentimes in the morning, when conditions are calmer, you see the effect. But we have, other bloom formers that also have mechanisms for slowing their fall. So things like diatoms often have little projections that stick off, and I'll show you some pictures in a second, which allow them to be more buoyant, have a higher surface area. Now, some algae that form blooms also produce toxins, and we already mentioned earlier this semester red tides, dinoflagellates. Fortunately, the Lingulodinium that we have off our coast here usually are not lethal, but we have a number of strains of dinoflagellates that grow in warmer waters, like on the East Coast and in the Gulf of Mexico, that do produce things like neurotoxins, which are lethal. And so oftentimes they have to close shellfish fisheries when there's blooms of these algae, because mussels and oysters and other shellfish will trap them in their gills as they're feeding on them. It doesn't hurt the shellfish, but if we eat the shellfish, then we would be affected. And so we can see these problems. Some cyanobacteria also produce toxins. They usually don't hurt people, but they're known to kill livestock in freshwater ponds and lakes where livestock might be drinking. And then we've also talked about how the high biochemical oxygen demand that arises when you have huge blooms of algae which die, fall to the bottom, and then are respired and decomposed, depleting oxygen and killing fish, as we've talked about. All right, so algal blooms play an incredibly important role in both freshwater and marine environments. And it's more, so what I want us to do is have a quick breakout session, and I want you to discuss in your group any ways you can think of to try and reduce the global incidence of algal blooms. So unfortunately, since we are running a little bit behind, we're not going to be able to do too long, but I'd like you guys to just come up with at least one or two suggestions for each group to reduce the incidence of global algal blooms. Adam, are you seeing the thing to join your breakout? Okay. Perhaps if we could have one person from each group maybe share one idea you guys talked about. So let's start with group one.
Yeah. So the group came up with one idea, which was the prevalence of biodegradable waste. So not just trash in general, but also like if it's like super biodegradable, that would contribute to like the amount of nutrients that are available for like algae to flourish. So reducing that would be one way to combat that.
Okay, as long as the biodegradable product doesn't release nitrogen or phosphorus into the environment, I guess. So you'd have to be careful about which, but certainly biodegradability usually plays an important role in reducing waste at least. So, okay. Group two, what did you guys talk about?
I think that may have been us. I'm not sure. I don't remember. But we did talk about limiting the amount of fertilizers that farms can use.
Okay. So, I mean, this is definitely something we want to try to do, but of course, a farmer is going to say, hey, you know, I need to grow crops so that you people can eat, right? So, um, you know, it's definitely one of those tricky issues, but certainly being more targeted, more efficient with fertilizer use. And I know some farmers are starting to use really high-tech systems to monitor nutrient levels and deliver it almost like at a plant by plant level, so that instead of just spraying huge amounts of nitrate fertilizer all over the ground. So getting more efficient that way is definitely would have a huge potential impact. I think you were in group three, Jocelyn. Group two was Ashlyn, Charlie, Janice, Jonathan, Lillian, and Rika. Anybody from that group?
Yeah, we were talking about a like a phosphorus locking agent. It's called Phoslock. It's derived from lanthanum. I guess it binds reactive phosphorus in the water, so algae can't use it.
Okay. Yeah, so it probably is a like a flocculating agent or something where it would fall to the bottom then and kind of be unusable by the algae.
Mm-hmm.
Yeah, and of course, if you can't solve the problem by stopping the source, at least you can maybe minimize the impact. One of the problems with phosphorus, though, is if it sits in the sediments over time, it can be leached back up. And so that might be a good short-term answer, but it may not last forever. But thanks, that's a great idea. Room four, that was Amy, Angel, Hector, Kevin, and Sandra. Anything you guys want to share?
We also mentioned limiting fertilizer use. Maybe
Okay.
if that's not super possible all the time, maybe they could find a way to limit the runoff from fertilizer into the oceans. And then we also talked about thinking about some kind of competition for the algae to prevent the algal blooms.
So competition with something else, like what? Or different types of algae? Is that what you were thinking? What?
We weren't really sure.
Okay. Well, you could maybe have a competition between like a toxic versus non-toxic strain or something like that, perhaps. Maybe you could engineer one that didn't have some of those properties. But one of the challenges out in the environment, though, is like setting up conditions so that it would favor one over the other might be difficult. But I guess another possibility would be to use aquatic plants to try and suck up some of the fertilizer. But that would be more for freshwater. All right, then group five, we had Janine and Jenny, Johan and Marwa.
So along the lines what Amy and you said, we thought about having physical efforts done on the water body, like actually removing the algal bloom, or by using some sort of non-harming chemicals or use of genetic bacteria, which help to eat up the algal bloom somehow and make it sink to the bottom in a way that's not harming the aquatic life.
Okay, cool. All right, well, those are all interesting ideas, and you know, there's no magic bullet. I just wanted you guys to kind of think about, because these are problems that we're going to be facing, and really your generation is going to be facing for many years to come. And so the more ideas we can get going, the better. So the last thing about algal blooms I just want to show is some pictures. So if you see sort of that pea soup green, that's often cyanobacterial, cyanobacterial blooms, but you can get blooms of these purple organisms, which are those non-oxygen producing or anoxygenic bacteria that are photosynthetic. This is the purples living aside right next to eukaryotic oxygenic phototrophs. And then I've used the term diatoms before. So we have different diatom shapes. So some of them form these discs like a roll of coins. Some of them form frustules like this. The disc ones look like this when you're just looking at a single one. They're very beautiful. In fact, during Victorian times, often there were groups of like particularly wealthy women that would get together and look through microscopes and look at diatom frustules for entertainment. I guess those were pre-television days, right? So, but this is a charophyte, which is actually kind of a relative of some early branching relatives of land plants, Euglena, that you've probably seen before. Dinoflagellates have this tripod shape. And then there's some simpler things like green algae and coccolithophorids. So the last thing when we're talking about the pelagic environment, which is related to marine snow but is not solely on particles, is the microbial loop. So traditionally, when I was learning, but I'm guessing in your grade school science classes, they still talked about food chains and food webs, yes? And so what is a food chain? What did you guys learn in like 4th grade science about what a food chain is?
Animals eating each other.
Yeah, so like little things being eaten by bigger things, being eaten by bigger things, right? And so the focus there is going to be on fish and larger invertebrates, things like copepods, which then eat algae. And bacteria are not really considered in those traditional food webs very well. But what we now appreciate is that microbes play an incredibly important role in cycling of nutrients in carbon, nitrogen, phosphorus, etc. And it's often not a linear, but more of a loop or a circular approach. So what you can see here, so this is kind of the linear food chain. Algae being eaten by zooplankton, being eaten by fish. But it turns out that like phytoplankton are these particulate organic material, right? They may aggregate as marine snow and they may fall out, right? Or they may get lysed and burst open, releasing dissolved organic material. And this could be because a virus infects them or because they get eaten, and not necessarily eaten by a large zooplankton, but they may be eaten by protists. And then that dissolved organic material may be taken up by bacteria, but the bacteria then get eaten by the protists. They also get infected by viruses, and they release the carbon and nitrogen and phosphorus and iron and things back into the water. So what you can see is the microbial loop tends to be circular and keep things away from the bigger, so-called bigger things. And so you have export that's not really considered in this model, and you also have this kind of like circle that goes around. And so this plays a very important role in terms of like where the carbon and nitrogen and phosphorus are. And it keeps the carbon from moving into higher trophic levels, right? So those small, very tiny primary producers, usually cyanobacteria, or there's a very their sister group called the prochlorophytes. These are very similar to cyanos, but they have a little bit different pigmentation. And so these are dominant, particularly in the open ocean, far away from land. So these picocyanobacteria end up creating long food webs, or they end up getting lysed by grazers or viruses before they can even be consumed by higher organisms. And so what this leads to is a loss of energy, making it to large charismatic animals like fish. Okay. So let's have our last poll question and our last slide of this lecture. Sorry. So which of the following would be a logical outcome of the microbial loop? So greater efficiency in carbon and nitrogen cycling or nutrient cycling mean that large predators have more food. Greater efficiency in the same means that large predators have less food. Because they are so large, sharks are unaffected by the microbial loop. Or D, none of the above. So let me relaunch the poll. So which of those four is the best answer? So large predators like fish, right? Or it could be sharks, orcas, tuna. Okay, go ahead and log your answer. Last chance. Okay. All right, so most of you put A and B, although 4 of you did say none of the above. All right. So it's coming down to this. D is not correct. One of these is correct. But it was a little bit tricky. So I think it's the way I phrase this in the sense of talking about efficiency in nutrient cycling, right? And we tend to associate that positively. But why is B the correct answer instead of A? Why would this efficiency or the loop cause it to be bad for large predators? So somebody maybe who chose B, why wasn't A the correct answer? Let's see in the chat. Robin said the microbial loop hoards nutrients, right? Yeah, so it traps nutrients and essentially it creates more steps. And if you think about it in a food chain or a food web, every step you have, you're going to lose some. In fact, it's often quite a bit. So if you have, instead of like 3 steps, like that simplistic phytoplankton, zooplankton, fish model is, if you have 10 steps or maybe 100 steps of it cycling in the microbial realm first, we're going to actually have less of that carbon and nitrogen and nutrients moving up to the large apex predators. So the best answer was B. Okay. So it may be an effective and efficient and healthy ecosystem, but it actually is good in the sense that you don't want too many apex predators out there because then you wouldn't have as many fishes, etc. It's all very delicately balanced, and of course humans are impacting the other end of things with overfishing of sharks and large predatory fishes, but that's not a topic for this class. You can take fisheries to learn about that. All right, so that is the end of that lecture. Any questions about the pelagic realm? All right, so we have about a half an hour to get started on extremophiles. So we will go ahead and get started on this. Obviously, we're probably not going to finish this, but, and then of course on Wednesday, we won't be having a lecture. We'll be having our discussion. So we'll finish this up next Monday. All right, let's keep it going and have another poll question, because why not? Let me see if I can get. All right, so in which of the following habitat would you expect to find extremophilic microorganisms? The International Space Station, a hot spring, farm soils, both A and B, or E, all of the above? Which do you think would have extremophiles? All right, log your answers. Okay. So it looks like everybody agrees that hot springs have extremophiles, right? And so then the question is, do any of these other two have extremophiles? So if you think A, if you put D, then you think A and B. So somebody who put D, both A and B, why did you choose that? Why are there extremophiles at the space station?
There's a lot of gases and extreme heat.
Okay. Well, so actually in space, it's really cold, right? So you're actually more likely to have, if you're in the space station, as long as you're if you're outside the space station, it would be really cold. When you say gases, what gases are you meaning? Like gases from space kind of thing, or?
I think I misunderstood it. I had put B, but.
Oh, okay. So we all know what the International Space Station is, right? So, you know, it's out in space, it floats around, you go up there, you can spend, 6 months or a year potentially and come back to Earth. And but it turns out that there are lots of experiments that are run on extremophiles in the space station. And one of the reasons that it's a good place to study this is because it's outside of Earth's atmosphere. And what do you guys remember the atmosphere does in terms of protecting us from extreme conditions?
Blocks heavy UV.
UV radiation.
Yeah, exactly. Thank you, guys. So ultraviolet radiation is really prevalent out in space, right? And so there's quite a few experiments that have been done. And incidentally, this impacts both UV radiation and gamma radiation impact the astronauts when they're up there, particularly gamma radiation. In fact, it's one of the major obstacles to sending people to Mars is the amount of radiation that they would experience between Earth and Mars, just getting there. And presumably you'd want to get back too, right? Because spaceships can't carry the lead shielding that would be necessary to protect against the gamma radiation because it's so heavy to get off the Earth's planet. So the space station actually has quite an extreme environment, but it's usually very cold, high UV. Hot springs, of course, very hot. So some of you put E though. Why would farm soils have extreme microbes? What was somebody who put E thinking on in terms of that?
I was thinking like extreme is relative, so they'd be able to withstand like a lot of the fertilizers that are being used, or like be tolerant. I was thinking the UV angle, like sun just hitting the soil constantly. So like there's
Okay.
no reason that they wouldn't live there.
Yeah, and I guess in terms of framing this question, there would definitely be extremophiles that you could isolate from a farm soil. However, the vast majority of organisms living in farm soils would not be. So the answer I was really going for here was D, but technically speaking, E would also be a correct answer as well from that perspective, Victor. So yeah. All right. So let's talk about extreme habitats. And oftentimes when we think of these, we think of the conditions, right? Remember those terms we used, ought ecology or ought ecological factors, things like conditions that aren't consumed. And so under high temperatures, of course, we know about thermophiles. The term that's used for low temperature loving organisms or adapted organisms are psychrophiles. High pressure, right? And that, as I mentioned a few minutes ago, in the deep sea, for example, or in the deep subsurface rocks, pressures are enormous. So the pressure goes up 1 atmosphere of pressure for every 10 meters of depth. So if you're at 3 kilometers deep, that's a lot of pressure. So the organisms that live there are called barophiles, or sometimes they're called pisophiles, and I'll give you a spelling for that later. High salinity, of course, halophiles, and the paper we're reading for Wednesday will cover that. So we'll be talking more detail about that. And then extremes of pH, right? So you can have low pH acidophiles and you can have high pH alkalophiles. So we'll start by talking about thermophilic habitats, right? And you can see right here, what's this a picture of?
Like a deep sea event.
Yeah, so we call these black smokers. These would be very high temperature vents. And they would be black because they have large amounts of iron and sulfur in them, which forms pyrite, which has is like a black precipitate. There's also, and so these can go upwards of 300 degrees Celsius. At lower temperatures, like upwards of 73 degrees C, you would see different chemistries. And so they would be called white smokers because oftentimes the fluids venting from those hydrothermal vents looks white. Now, 73 degrees is still very hot, right? If we put you guys, if we were put at 73 degrees, we'd be dead, right? That's double our normal body temperature, okay? But from a microbial standpoint, it's kind of, minor, right? Because if you can live in 350 degrees C, it's all relative, I suppose. So one of the cool things to think about in this picture is that this water may be like 300 degrees Celsius. And right here, this little bit of water over here is 2 degrees Celsius. So what this does is it sets up an amazing gradient between the superheated, super mineral rich, iron rich, anoxic water, typically anaerobic, whereas this is cold, nutrient low, uh, and oxygenated water over here. So these gradients are exploited. And so that's one of the reasons we have sulfur oxidizing chemotrophs that love to live in habitats like this. And they use the oxygen from the seawater and the sulfide from the vent water for their metabolism. Anything living down here would need to be a thermophile if it's living near this water. And it would also be under high pressure as I mentioned, 10 meters, 1 atmosphere, so over like 300 atmospheres. There are other thermophilic habitats a little closer to home, and these are just a selection of pictures from some of my own research in Yellowstone Park. So things like boiling pits, mud pits or mud pots, they're called. I've already showed you some pictures of some of these hot springs where there's microbial mats that form. And then this is a geyser, right? So you guys have all heard about Old Faithful or similar geysers, right? Has anybody here ever been to Yellowstone Park? Well, when COVID is over and we're able to travel, it's an amazing place to go. It has all of these amazing habitats, as well as all the wild animals. And in the summer, it's just beautiful, as you can see from these pictures. But in the winter, there's like lots of recreational things, snowmobiling, cross-country skiing, and it's far more crowded in the summer, of course. It's like Yosemite, far more crowded in the summer. But from our perspective, though, what's important is that Yellowstone Park sits atop a massive like chamber, a magma chamber. And so Yellowstone has the greatest concentration of thermal features on Earth. So that's part of what makes it such a wonderful place to go do research. Now, some of these will also have other features in addition to the high temperatures. So you can get pools that have pH down below, down to near zero. And I'm going to show you a picture of that later on. So thermophiles that live in these environments, whether it's at a deep sea vent or in a hot spring or wherever, can be categorized by how high they can go and how thermally loving they are. So 50 to organisms that grow in optimally from 50 to 80 degrees C. Again, hot, but not as extreme, are called thermophiles. Anything over 80 C would be a hyperthermophile. So those organisms like we talked about that live in autoclaves at 120, which is like the record for cultured organisms, would all be hyperthermophiles. Now, which kind of microbes are these? Well, typically for all of these extreme conditions I'm going to talk about today, it's usually an archaean that has been cultured that is the record holder, so to speak. So for example, the 122-degree organism that grew in those autoclaves was Methanopyrus, an archaea. This is an anaerobe and a chemolithoautotroph. Okay, but not to be outdone, the bacteria, they may not go over 100 C, but there's some really high temperature stuff. And in fact, this Aquafex is very common in some of those hot springs I was showing you in Yellowstone National Park. And they also are chemolithoautotrophs, sulfur chemolithoautotrophs. They're also found in deep sea habitats. Thermos. So who has heard of Taq polymerase? What does that do?
It's used in the PCR.
That's right. So Taq polymerase is short for Thermus aquaticus polymerase, right? So it's the DNA polymerase that came from Thermus aquaticus, which is a thermophilic bacterium that was isolated in Yellowstone National Park decades ago by Tom Brock, a very famous microbiologist from the University of Wisconsin. He cultured this organism, and at one point he was asked by somebody if they could borrow or have a clone, a batch of his Thermus aquaticus, and he gave it to them freely. Incidentally, he isolated it freely from Yellowstone Park as well. Well, since then, billions and billions of dollars have been made using Taq polymerase that was originally isolated from Thermus aquaticus. None of that money came back to Tom Brock. None of that money came back to the National Park Service or the federal government. So now when you do research in places like national parks, you have to do elaborate permitting. And there are sort of standard agreements where you will give 50% of all royalties that might result from the biotechnological development of a product from anything in the park to the federal government. But it was too late for TAC polymerase. We can't get that money back, but that's sort of the Tom Brock TAC rule. And so it's much harder to do research in national parks now than it was when even I was first starting there. The annual permitting and reporting that's required is quite elaborate. But in this case, there's a pretty good reason. I just like to share that because it's like, oh, if only I had the patent to TAC polymerase. It's owned by a company, a Swiss company now, but it's changed hands a number of times. Incidentally, you can get your own culture of TAC polymerase or TAC thermus aquaticus and isolate its enzyme for your own personal use for free if you have the technical know-how to do it. And you don't have to buy it. But anyway, most people don't bother. So the opposite end of this, instead of thermophily, would be psychrophily, cold temperature loving organisms. Now, unlike with high temperatures, many organisms are not true psychrophiles. Many of them are psychrotolerant and would prefer to be at higher temperatures, but there are some true psychrophiles as well. And where do we find these? Of course, we're going to find them in super cold places like Antarctica, in the sea ice at polar regions, right? If you isolate bacteria or algae from those habitats, they're likely to be very cold temperature adapted. The deep ocean, as we've said, is about 2 degrees, right? So much of the Earth's volume is filled with organisms that are likely going to be cold adapted, psychrophilic, or at least psychrotolerant. And then, of course, as we've said repeatedly today, they also have to tolerate what in the deep ocean?
Pressure.
Exactly. So oftentimes you'll see psychrophiles that are also barophiles in those habitats. Now, if you're a psychrophile, you have to have a temperature optimism below 20 degrees C, right? Below room temperature, so to speak. And in some cases, there are organisms that have temperature optima below 10 degrees even. But from a thermodynamic standpoint, it's really hard to make a living if you're going to be optimally adapted to grow at like 8 degrees Celsius. Why is that? What do you guys know about sort of physiology and enzymes and things like that?
Wouldn't the chemical reactions not be as like quick?
That's right. So, Michaelis-Menten at low temperatures, right? The curve is on the left-hand side. It's not going up. So adaptation to those low temperatures, it does happen, but it would really only be under certain circumstances. Far more organisms are psychrotolerant, meaning they grow optimally above 20 degrees C. However, if they are placed at 10 or 15 degrees Celsius, they can survive and grow just at a lower rate. So what we're really looking at here is where you're optimally growing. And so this makes kind of a lot of sense. But there are still plenty of psychrotolerant and psychrophilic organisms out there. Okay, so we got thermophiles, we got psychrophiles, and how do these organisms adapt to this? So one of the things we have to worry about are lipids. Why do we have to worry about lipids when we're thinking of temperature? What do you guys know about lipids and where are they found inside the cell?
Is it the membrane, the casing of microbes?
Yep, so they're going to be, right, the lipid bilayer of the plasma membrane. So why do we care about temperature when we think about lipids? let's see in the chat. Angel says fluidity. Yes. So remember the fluid mosaic model of lipids, right? They're not static. They're kind of undulating and moving, and they need to stay fluid so that their proteins that span work properly, so that things can diffuse, and so the membrane doesn't gel up or lock, right? And so maintaining fluidity without it getting too fluid is a real challenge, especially for thermophiles. So for thermophiles, they tend to have saturated fatty acids, right? With longer chains, okay? Right? So you've got your double membrane, right? And you've got your chains that are dangling in between. You've got the polar heads and the hydrophobic zone in the middle where the fatty acids are. But if you have longer chains and you saturate those fatty acids, it maintains structure. Under high temperatures, your lipids want to fly apart, okay? Under low temperatures, they want to lock up and freeze. So thermophiles tend to have saturated longer chains. Psychrophiles tend to have unsaturated fatty acids with shorter chains so that they can stay fluid as long as possible as the temperature drops. So if we look at this just as a reminder, right, saturation, right, just means you've got protons on all of them. You don't have those double bonded carbon-carbon bonds. So thermophiles are going to tend to be in this category, okay? Saturated. Psychrophiles are going to have unsaturation and shorter fatty acids. Another thing that has been associated with, and it makes a lot of sense in theory, but may not be as important as it seems, is that the GC content, if you increase it, that can increase the stability of DNA in thermophiles, okay? But one of the issues with this is that if we look at GC content in thermophiles, they don't all show this, right? So when we talk about GC content, what I mean is if you add up the numbers of A's, C's, G's, and T's, the higher the numbers of G's and C's versus adenine and thymines would result in a bias. And so that would be over 50% G's and C's. And why does it make sense that you would have more G's and C's at higher temperatures? Who remembers their biochemistry here? What is it about those G's and C's in terms of their bonding that differs from A's and T's?
Their
Is it
hydrogen
because
bonds?
they
Hydrogen bonds.
And how many bonds do they have?
3.
have
3.
That's right. So there you've got 3 bonds instead of 2. So it makes a lot of sense in a theoretical setting, as I said. But when we go looking at thermophiles, not all thermophiles have high GC content. And there are even low temperature adapted organisms with high GC. So it may not really be. Certainly something that's more important, and this actually should be moved over, I apologize, is the importance of proteins. So heat shock proteins, which are actually just stress proteins, but they were first identified in E. coli exposed to heat shock. So that's how they got their name. And these are chaperonins, right? You guys have learned about chaperone proteins before. What do they do? Who remembers from biochem or
They're like
genetics?
spectators during DNA replication. They aid in the DNA replication.
Yeah, so they aid in a lot of processes. So basically, these are the proteins that take other proteins that are misfolded or unfolded and put them back together again. So chaperone proteins often aren't the enzymes that are catalyzing. They're making sure the enzymes that are catalyzing a reaction are in the proper shape or conformation to make it work. And so this is likely very important in these extreme conditions because high temperatures, for example, would cause enzymes to denature or open up, right? So heat shock proteins likely play a very important role. And so when we're looking at enzyme activity, I alluded to this a little bit, is that under cold temperatures, you're going to tend to have low enzyme activity. Under high temperatures, it will go up. So if you're a thermophile, you have the advantage that you're functioning under like more favorable conditions. However, at very high temperatures, those enzymes are going to want to fall apart, even if chaperones have been produced. And so we measure the activity of enzymes using the Q10 value. You guys learned about that in any of your classes, your chem classes, maybe talk about Q10 values. So that's the change in the activity of the enzyme for a 10-degree temperature difference. So typically you end up seeing an increase in the temperature activity as temperature increases, but then it hits a threshold and it crashes. So these enzymatic changes are going to be really important. And this is often what evolves, right? So you have mutations, you have amino acid changes, which change the proteins and those enzymes. And so over time, under high temperatures or low temperatures, the enzymes become adapted and sort of tuned so that they function optimally under the conditions that that organism is living under. And that is one of the things that allows those organisms to thrive and survive under the conditions. Does that make sense? Any questions about these temperature adaptations? All right, let's see. Might be able to get through this. So under high pressure, we said they were called barophiles, but there's also this term piezophile, which we use interchangeably in this class. And so I mentioned that in the deep sea, we often have psychrotolerant or psychrophilic organisms. And many of these are also going to be barophiles or piezophiles. We do have some thermophiles that live in the deep sea. And where would those be living? Where do we see thermophiles in the deep sea?
The hydrothermal vents?
Exactly. So like what I showed you a few minutes ago. But the vast majority of life in the deep ocean would be barophilic psychrophiles or psychrotolerant. And so one example, this is an organism Colwellia that was isolated from sea ice. But it is found in the deep ocean as well. And they're often barophilic. I mentioned that if you're a temperature adapted, you need to have fatty acid composition changes. Oftentimes we see the same thing here with pressure. So it turns out that under high pressure, your lipids are going to want to lock up again, just like we saw with the psychrophiles. And so just like with psychrophiles, unsaturated and shorter chain fatty acids help maintain fluidity. So oftentimes those psychrophilic, barophilic microbes will have the fatty acids be unsaturated, and that benefits them from both angles. They also have adapted proteins that can function under these high pressures as well. Now, one of the things to think about in terms of cultivation is that if we're going to try to culture these organisms, if you go down a kilometer and send a bottle down, let's say, and you figure out a way to get that bottle to close, sample some seawater, and you bring that up to the surface, pop it open, and try to culture something from that water, why would that be a problem? Why do you think it'd be a problem if I just popped open this thing from a kilometer down and then tried to do like streak plating on a plate or something with it.
The conditions are completely different. For example, the pressure.
Right. So if the pressure is going to be totally different, what do you think would happen to those cells? You're absolutely right, Jocelyn. But what would happen to the cells, do you think?
They would burst.
They would, yeah, they would lyse or burst. Exactly, Carlos. So we need to use things like isopiastic sampling devices, which is basically a fancy way of saying pressure chambers, right? So we would send a pressure chamber down, close it up, grab that water, bring it up, and keep it pressurized. And then when you culture it, you would need to culture them under high pressure as well. That's a lot trickier than just grabbing samples. You won't be surprised that the earliest studies were done by just grabbing samples, opening them up, and see what grew. And lo and behold, they didn't get a lot of barophiles out of those. So using these new devices makes it essential. And this is particularly true if they have gas vacuoles, which will blow up. But really, almost any organism adapted to the high pressures will need to stay at some pressure. Interestingly, if you did that on the deck of a ship where it was sunny and warm, right, you'd also probably kill those organisms because they need low light and low temperatures as well. So you've got to be really mindful when you're trying to culture these organisms. All right, so I want to do this last poll question and then we'll stop, but this is while it's fresh in your minds. So which of the following adaptations would you not expect to see in which would you not expect to see in a polar deep sea bacterium? Okay, go ahead and log your answers. All right. So it looks like most of you put D, saturated lipids. So why did we say saturated lipids? Somebody who put D, why is that not what you expect down there? Because what did the saturation, where did we see saturated lipids? What group?
And thermophiles.
Right, and why does the saturation help thermophiles?
Maintain structure.
Right, so it makes it stiffer if you think about it, right? So the saturation makes it stiffer. And so we want to be fluid and maintain things as fluid as possible if you're cold and in the deep sea. So they would have unsaturated short chain fatty acids in their lipids. And the chaperonin proteins, even though there's heat shock proteins, there's also cold shock proteins that help. So there's really going to be all A, B, and C. So D was the correct answer. So good job, guys. I know it's hard to remember which is which, so but you need to try and keep it straight. All right, so we will go ahead and stop there, and we will pick up